Okay, so hey everybody, my name is Raghav, and I'm a freshman at Carmont High School in Belmont, California. And my topic is about the usage of induced pluripotent stem cells in sports for muscle injury treatment. So in sports, an athlete's worst fear is facing an injury. Today, we will be focusing on muscle injuries. And with muscle injuries, there are three main problems. For an athlete recovering from an injury, besides, how is my injury doing, their topmost priority is when can I get back to playing? Unfortunately, for current athletes, when it comes to muscle injuries, the answer to that question is not very soon. Second, I noticed that muscles, especially those in the lower body, are the most exercised part in competitive professional sports. They serve multiple functions, which only emphasizes their importance to an athlete more. Finally, I came to understand that even when a muscle is healed by the body, the scarring that occurs during the healing process can often weaken the muscle, leaving it susceptible to further injury or damage to other structures, such as tendons or bones. As I saw that stem cells had been an emerging topic in the field of biotechnology, I was led to the question: how can induced pluripotent stem cells be used to accelerate the recovery of muscle injuries that occur in sports? To answer this question, we can condense our whole research process into three main steps. First, we can turn regular cells into induced pluripotent stem cells. Next, differentiate these regular induced pluripotent stem cells into skeletal muscle cells. And finally, implant these cells into the ruptured, torn, or strained muscle area to help heal it. Oh, sorry, wrong side. So the first step involves creating an induced pluripotent stem cell. An induced pluripotent stem cell, or iPSC, is a cell that can differentiate into any cell in the body. So if you have an induced pluripotent stem cell, it can differentiate into a neuron, a muscle cell, a liver cell, anything really. In addition, an iPSC can be derived from any cell in the body. So you can take any cell in the body and turn it into an iPSC. You can also take that iPSC and turn it into any cell in the body. So it's a very versatile cell. To derive an iPSC, the usage of the Yamanaka factors is the most common method. The Yamanaka factors are the 4 genes that define a stem cell. By applying these 4 genes through CRISPR-Cas9 editing, we can turn any regular cell into an iPSC.
Awesome. Let me just pause you there for a second. Our view is still just on the very first slide.
Oh.
And so I just want to make sure we aren't missing anything.
I think the very first slide, so it's on the title slide.
Yes.
Oh, okay, that's not good. No. Can you hold on? What about now?
Oh, good. Okay. Yes, they're moving through. Now we're on the research question.
Okay. I think. All right. Does it say step 1, create an iPSC?
It says research question: how can induced pluripotent stem cells be used to accelerate the recovery of muscle injuries that occur in sports? Okay, now we're on the step 1.
Okay. Sorry about that.
No, don't apologize. All right.
Okay. So, okay, yeah. So once we have an induced pluripotent stem cell, we have to differentiate it to make it a muscle cell. Although there are many different methods to doing this, the most common is to just mimic the development of an embryonic stem cell. To do this, cell signaling pathways are most commonly used. And the most common for differentiating into a muscle cell is the WNT or Wnt signaling pathway. And when I mean mimic the process of an embryonic stem cell, I mean all humans started as embryos, right? So in those embryos, there are embryonic stem cells, and those embryonic stem cells are what gave rise to like the first specialized cells in your body. So actually, this chart right here on this chart is it's a chart about the different pathways that any pluripotent stem cell can sort of travel, I guess, um, to give rise to different types of cells. So in our case, for a skeletal muscle cell, it would have to commit to the mesodermal lineage. Just a side note, all cells can be traced back to either an ectodermal, mesodermal, or endodermal lineage. In the case of muscle cells, they start by committing to the mesodermal lineage. Then they go to a paraxii mesoderm. Then they form, and finally they become skeletal muscle cells. Mo most times, more than one cell signaling pathway is required to be able to differentiate into a fully specialized cell. And so it's a very complex process that can be somewhat challenging.
Awesome. Just so you know, we're still on the step 1 slide.
Sorry.
No, it's totally fine. And if you want to send me your presentation and have me share and flip through, I'm happy to do that too.
Can you see it now?
Yes.
Okay. Should I okay. Okay, yeah. So anyway, what I was saying was all pluripotent stem cells, they have to go through one of these, they have to commit to one of these 3 stages at some point. So if for a iPSC that is trying to differentiate into a skeletal muscle cell, it must commit to the mesodermal lineage first, and then it'll differentiate further into a paraxii mesoderm, and then it'll become a somite, and then it'll finally differentiate into a skeletal muscle cell. Once we have the muscle cells, we have to, the next step is to implant them into the injured area such that they are able to heal the injured muscle effectively. To do this, we have to make sure that these muscles in this injured area can first divide and reproduce, and second, they stay confined to the site of injury, so they don't just go wherever they please. This can be achieved using a fibrin gel and a thrombin solution. What that is basically, so you'll take muscle cells, you put them in a fibrin gel, and then you can either surgically implant this fibrin gel into the site of injury, or you can inject it because it is fairly, it's like a liquid sort of. And then after that, you would inject a thrombin solution. And what this does is it hardens the fibrin gel enough such that the muscle cells inside that fibrin thrombin solution, they stay confined to the site of injury, so they don't go like roaming and go off. This figure that I have below, it's from one of the papers that I used for my research. It shows 3 different methods of healing injured muscle tissue. In this box, you can see they just left the injury alone and let the body treat it. For this one, they only placed gel, the fibrin gel, and then for this one, they did a mixture of fibrin gel and muscle cells. In this control box, you can see there is a clear scarcity of cells in this tissue. You can see and the same goes for this one. It's a little bit better in the gel, but the muscle tissue is very weak in general. Over here, though, you can see the tissue, first of all, there's a lot more cells. It's a lot more populated with cells. Also, you don't see much imperfections within the tissue. There aren't tears and gaps like the other two boxes. And also, if you notice that there are blue cells that are dyed, those are actually the cells that were implanted with the fibrin gel. The red ones are just the pre-existing cells that I guess did not get affected by the strain or tear. But if you can see this, you'll see that around 40 to 50% of the muscle tissue is from these these cells that we derived ourselves and placed at the site of injury. And what that shows is that, you know, the body, as wonderful as it is, it has its limitations. And although many of us assume that, you know, you tear a muscle, it'll grow, it'll regrow, and you know everything will be fine. That's actually not true at all. And methods and processes like this should be required to fully restore the function of the muscle. At the end of the day, it's all about, did you answer the question that you had at hand? You know, if we just discussed all of this and none of it's applicable, then, you know, it really does not matter. So I concluded that using induced pluripotent stem cells to treat muscle injuries, firstly, accelerates the recovery process, helping athletes get back to playing sooner. And also, it results in significantly less scarring of tissue, reducing the muscle's susceptibility to injury, as well as other structures like tendons and bones. In the future, I would definitely like to focus on less sports, a little bit more stem cells. I would like to focus on whether or not iPSCs can be used to retain the function of the nervous system for those suffering from paraplegia. I've always been interested with like these kind of things. I really I think the nervous system is a very interesting aspect of science, and it's yet to be fully like discovered. And I really want to see if stem cells can be applied to that. And yeah, that's it. Thank you for listening. Any questions, I'm completely willing to answer.
Wonderful. Thank you so much. That like visual that you shared on the slide around like implants in the muscle cells and gels, it was like so cool and also kind of gross and like, oh, like very haunting. I'm kind of obsessed with it now.
Yeah.
There is a question in the chat. I just have to ask out of my own curiosity first. Have you yourself experienced like any sports injuries or like a muscle tear or anything like this?
So thankfully not yet. I've broken a
Knock on
few
wood.
bones, but yeah,
Oof.
knock on wood.
Oof.
But actually, one of the reasons I didn't really focus too much on bones and tendons was because with induced pluripotent stem cells, obviously it's a cell, right? And you're differentiating into a cell. And I realized that tendons and bones, or at least tendons, they're not very cellularized. So it's very hard to apply cells to that. So it's really kind of like pointless if you're applying induced pluripotent stem cells to things that don't really have many cells. So that's mainly why I chose muscles as my topic. But yeah, no, I have not experienced any muscle tears or things like that.
Excellent. Yeah, hoping that continues to be the case.
Yeah.
Awesome. Well, yeah, you can read them too, but I'll read them out loud just so everyone knows the questions in the chat.
Mhm.
Some studies say that autologous iPSCs can still trigger immune responses. And so with your research, what is going on if you take a look at the molecular level, and would gene editing be the way to fix this? Also, how realistic would it be for program iPSCs to favor fast twitch or slow twitch fiber regeneration?
Yeah. Okay. Wow. That's a big question. I think So the first part was about what is going on if you take a look at the molecular level, and would gene editing be the way to fix this? Uh, um, to be honest, the project isn't really focusing on what's happening at the molecular level. It's really about trying to create like a lot of them. So um, I'm not exactly sure about what they mean by gene editing being the way to fix this. Um, can still trigger immune responses. Gene editing be the way to fix this. I'm not exactly sure what they mean by that. But second part, how realistic would it be to program iPSCs to favor fast twitch or slow twitch fiber regeneration? Fast twitch or slow twitch fiber. I mean, I guess you could, but one of the things that I encountered while researching about iPSCs is that normally when you find a shortcut with induced pluripotent stem cells, it's there's a lot of different like after effects that happen, which is why most of the processes that are part of my methods are normally like the very direct long way there, because if you want to prioritize function instead of speed, then you should taking shortcuts with induced pluripotent stem cells isn't exactly the way to go, right? And um, yeah, that's mainly what I would say. But if you want to prioritize speed, then there's always different methods like overexpressing transcription factors, such as for muscles, I know it's like MyoD and MYF5. Those are definitely valid methods, but they can have a lot more side effects than regular methods.
Awesome, awesome. Yeah, I'm just like fast twitch and slow twitch for me just means, is there lagging in my Twitch streaming while playing a game or something. So that's amazing. Um, Another question for you. How difficult is it to produce an iPSC? Is it easy to produce the transcription factors needed to turn somatic cells into iPSCs?
Yeah, so in terms of like producing the iPSC, I mean, obviously you need a lab, but it isn't impossible. It's not that hard, but I would say there is some level of expertise needed to
Mhm.
understand, because they are very fragile and brittle. Because like even a little bit of variation in like the environment that they're produced in, um, if you like you have to make sure that the environment and the culture that they're raised in is stays the same throughout. You can't vary it, otherwise they're gonna have interesting results. So I'd say, yeah, and I talked about this. Is it easy to produce transcription factors needed to turn somatic cells into iPSCs? Yeah. So when you are differentiating induced pluripotent stem cells, when they're going through different stages of um, differentiation, they will produce transcription factors or markers themselves. So it's not too hard. It's pretty easy to understand the transcription factors. Need to turn somatic. Yeah, I guess you could use transcription factors to turn somatic cells, but like I said before, you overexpressing transcription factors is not the way that I would go. Um, I would try to take it through the whole embryonic stem cell differentiation process instead of just overexpressing one transcription factor.
Okay, nice.